I'd like to introduce Maria Colomé-Taché. She is a professor of functional genomics at the LMU. She's also a group leader in computational biology at the Helmholtz Center for Computational Biology. Her work focuses on single-cell epigenomic analysis, and I hope she's going to tell us a little bit about that today. Just briefly, Maria got a PhD in theoretical physics in Paris, did a postdoc in theoretical physics in Hannover. Then she moved into biology at the University of Groningen and stayed with biology ever since. So with that, I'd like to welcome her and wish you all the best. I have to make you co-host, right?
That's what I was going to say.
But if I log out, what happens? Can you manage everyone, or should I make you host?
Make me host if you can, then.
Okay. 1 second. There. Wait, there. Yes, you should be host now.
Yep.
Okay. See you later.
Good. Thank you very much, Frank, for the introduction. I would very much appreciate if some of you could share their screen with me. I mean, their picture, you know, such that I can see some faces where I'm talking and not just, you know, black screen. Excellent. Thank you so much. That's much better. Good. Let me look for my presentation such that I share the right screen with you. Wait a minute. That should be the one. I have a few presentations open and I have to make sure it's the right one. Good. You should be able to see my screen, isn't it? Full screen. I unfortunately have your faces on top of my screen, but it's great. So I will sometimes, you know, we need to move your faces around. That's all. Let me see if I can. Can I? Well, okay, whatever. I have no pointer. Do you see my mouse? Yeah. Okay, great. So I am the second speaker of this seminar series, and my job is to tell you a little bit more about the components of the epigenome. And then I will go more into my own research that has to do with single-cell epigenomics. So this is my group. I want us to start by that, such that you know who does the work, at least the work of the second part of my presentation, that it's going to be about our own work. We do computational epigenomics, which means that we are all very much interested in the epigenome, but the majority of us are non-biologists. So we have a big mix of backgrounds in my group. We've got a few people that are, you know, like more computer science or electrical engineering. We have people that are biologists that turn into programming. We have a pharmacist, we have a physicist. We have really like a very large mix of backgrounds. And we all kind of, you know, have a thing for programming, and we like to kind of solve biological problems with our computers. So I will start by telling you a little bit about just epigenomics and one of the main components of epigenomics that we can measure in the lab, and that will then inform us a lot about cell identity. And then I will go into my own work that has to do with single-cell epigenomics, and I will give you two examples of single-cell epigenomics, one of them with single-cell DNA methylation and the other one single-cell open chromatin, and then just one slide on single-cell multiomics. Oops, wait a minute. There we go. So epigenetics, going beyond the genome. I'm sure that you are all aware of the fact that, you know, the human genome has been sequenced. We have since many years now known very well the complete genomic sequence. So we know, you know, the letters that form the DNA of a human. It's very long. It has more than 3 billion bases. So, you know, it's a lot of letters one next to the other. And we do not just know the letters, the sequence of nucleotides that form our genomes, but we also know where are the genes located, and we have a pretty good idea of what these genes are doing. We have a pretty good annotation of the human genome in terms of, what are their genes and what is their function. Now, the question, though, is the following. Well, once we are done, we have the sequence of the human genome, and then we understand where the genes are. But, well, all of the cells in our body have exactly the same DNA sequence. So the question is, why don't we look like a blob? If, all of the cells are the same, all of the cells have exactly the same genes. What makes us not look like a blob, but look like a human that has more than approximately like 200 different tissues that perform very different functions. The tissues are made themselves or many, many different cell types, and these cell types are very, very specialized. So how is it possible that from one and the same DNA sequence, we are able to make so many different cellular phenotypes that will perform so many different functions? This is a course about epigenetics, so I guess it is no surprise that the answer is the levels of gene expression. So from the DNA to the RNA, there is a layer of regulation that makes it such that from one and the same DNA, we make different levels of RNA. And then there is even some layers of what we call post-translational regulation that make that from the RNA, we make different types of proteins. So the key here is that these different cell types that are part of a multicellular organism, we know that they are very different, both in structure and in function. And this is because these cell types, they become different from each other because they synthesize and accumulate different sets of RNA and protein molecules without changing the DNA sequence. And I was not here last week, but this point here, without altering the DNA sequence, is a point that was very much studied, like, around 50 years ago, where people didn't know, are we actually losing genes along the way? And, like the cells that have a particular function have actually lost a given set of genes, or do they become stable? And it's just that then they become on or off. So this is just like a very brief scheme where you would have a cell in a given tissue A. And when I put this scheme here, this black line always represents the DNA. And this arrow means that there is a gene at that location. And here, this gene is making an mRNA, while here, this gene is not making an mRNA. Now, all of this process of going from the same DNA to producing so many different types of RNA is regulated by the epigenome. So epigenetics is defined as the study of these stable changes in gene expression, which are not caused by changes in the DNA sequence. The DNA sequence stays always the same. There are no mutations. There are no genes which are, like clipped out of the DNA and they do not exist anymore. The DNA stays exactly the same, but what changes are the gene expression. So the expression of the genes. Now, wait, I have to move your faces again. As you know, epigenetics is a little bit of a hot topic. It was even more of a hot topic a few years ago, especially like in the media, like why your DNA is not your destiny, like this idea that with a given lifestyle, with a given nutrition and so on, you can actually, so to say, change the expression of your genes. And therefore, DNA is not everything. Now, of course, since the epigenome regulates gene expression, it is absolutely essential for organism function. And we know that if we get cells that have, let's say, a screwed up epigenome, there are many, many possible illnesses that appear, for example, cancer. We know that if we change the epigenome during early embryonic development, most likely the embryo will not develop, etc. So it's really very, very important for the functioning of the organism. And it is associated with illness because we will be producing too much or too little of a given protein. I guess all of that you more or less saw last week from a more like historical point of view. The epigenome is responsible for gene regulation. We can study it, etc. But the point of today is more, what are the epigenetic mechanisms? So what are, let's say, what is the molecular basis of the epigenome? And I will be talking about three of the most well-studied epigenomic regulation mechanisms, which is post-translational histone modifications, chromatin architecture, and DNA methylation. There are many other ways that we can regulate to produce from the same DNA different RNAs, different proteomes, but I will not go into those because they are more far away from my expertise, and some of them are also less well studied. So I will start with the organization of the DNA. As I guess you know, the human genome has 46 chromosomes, which means that every one of your cells, nearly every one of your cells, some cells have lost their DNA material, but the ones that still have the DNA material, every one of your cells has these 46 chromosomes inside of it. And if you were to take these chromosomes from inside of a cell and you were to stretch the DNA helix of, all of them, one next to the other, that would be 2 meters long, which is really long. It's really inside of every one of our cells, we have 2 meters of DNA. And the caveat here is that the cell nucleus is only 6 picometers in diameter. So it's really tiny, the nucleus of every one of the cells. If you think about it in analogy, this would be equivalent to packing 40 kilometers of a thread inside of a tennis ball. Yeah, so just to give you an idea, 40 kilometers is from the north campus of TU in Freising to the south campus of the LMU in Großhadern. So this is what 40 kilometers is, and you get this thread and you need to put it inside of a tennis ball. So it's a little bit of a tough job. And of course, we have some mechanisms to do that in the body. So how does this work? So how do we do to condense these DNA molecules very, very, very tightly inside of the chromosomes? So first of all, as you know, the DNA is a double helix, so it's already kind of old. But this double helix on the first level of packaging, the way it works is that the double helix is packed around some histone complexes, which have this kind of cylinders, let's say. So it goes around them, then it goes out and it goes around the next one, and then it goes out and it goes around the next one. So this is the first level of packaging of the DNA. And this is what we call nucleosomes. Then these nucleosomes, say that because here it says nucleosomes. So these nucleosomes are then kind of put in a kind of a zigzag structure that makes them become even more packed. And the zigzag structure form then these kind of loops that are very, very much condensed. And this kind of condensation is what I guess you've seen sometimes in pictures where people have, imaged the chromosomes when they are in mitosis, so that you have these very condensed chromosomes that have this very nice X kind of picture. So as a net result of all of this packaging, what we get is that the mitotic chromosome, this very packed chromosome, is 10,000 times shorter than this original 2 meters of DNA. And some of these structures can be observed in the microscope. Here is a pretty old picture already that shows you this level here of condensation. And this one here where you see this kind of, like a pearl necklace, let's say. This pearl necklace here is actually these nucleosomes. And the thread in between is the DNA. I will concentrate on this structure here, because this is the one that for an epigenetic regulation point of view, it's really important. So this structure here is what we call the nucleosome. So the nucleosome, I will move your faces again, sorry. The nucleosome, which is one of these units here, is made out of 8 histone proteins, 147 base pairs of DNA that is wrapped around these 8 histone proteins 1.7 times, so not really 2 times, 1.7. And this piece of linker DNA, which is in between the nucleosomes. As we've seen, they are responsible for the first and most basic level of chromosome packing. And this is how this is done. So every one of these units here is made out of this histone core and this piece of DNA. And the histone core, we can put it apart, and it's made of 8 histone proteins, which are 2H2A, 2H2B, 2H3, and 2H4. And histone proteins are extremely conserved proteins, and we have a lot of them in every cell, like, 60 millions, let's say, per cell. Now, the relevant thing here is that these histone proteins, so again, you see this kind of octamer here, they have this N-terminal amino acid tail. So these tails are made out of protein also. And these tails that kind of stick out of the main histone core, they can be chemically modified. There are many different modifications that can affect these proteins here. We call them marks also, but these are modifications. So, acetylation, methylation, these are probably some of the most frequent ones, but phosphorylation, ubiquitination, sumoylation, you name it. There is really a lot of different chemical modifications. And these chemical modifications can be in different residues, in different amino acids of this tail, and they can be in different tails. So, every one of these nucleosomes can be, decorated by a given number of chemical modifications that may be positioned in different positions of these tails. And this idea gave rise to this idea of the histone code. So why is that? So we know that these modifications, which are deposited in the nucleosome, are important for the regulation of the DNA, which is in that position. We know that it regulates transcription. It decides whether the DNA is transcribed or not transcribed in that region. It regulates the condensation of the DNA. It regulates whether the DNA is very packed or the DNA is very open. It regulates DNA replication. It regulates DNA repair. It regulates so many different things. And how this regulation is working is mainly kind of indicated by these different sets of modifications that can be put in the tails of the histones. So the idea, let's say, of course, it's very naive, but would be like there is an underlying histone code that we don't really know yet how it works. But if we know that there is a given combination of histone marks, then we would be able to say, aha, that position in the genome is being replicated. Or if we find another combination of histone marks, we could say, aha, that position in the genome is being transcribed. Yeah, things like that. Now, what is important is that, of course, these histone modification patterns are very, very, very well maintained. So there are many enzymes and so on that will go and put these modifications, remove these modifications, etc. And yeah, there is this, it's a little bit of a naive idea, but just to give you an idea that this histone code determines how and when the genes are expressed. Of course, as I mentioned, it's very important that histone modifications are inherited upon cell division, because if a histone modification at a given location gives us information on whether a gene is being transcribed or non-transcribed, well, if when the cell divides, all the histone code gets lost, then, like a skin cell that starts dividing will forget that it's a skin cell. So it's super important that these histone modifications are preserved upon cell division. Well, of course, it's also important that they get destabilized from the DNA when the DNA is replicated. So first of all, I think I have a picture. Yes. So, oops, sorry. So if you have here, you see your DNA is this red thread, and in yellow is this histone octamers. If I want to start replicating the DNA, first of all, I need to somehow, destabilize the DNA histone pairing, okay, such that the replication fork can actually access the DNA and start replicating it. Then, sorry, I need to move your faces again. After the DNA is duplicated, we have the double amount of DNA. So we need the double amount of histones. So this is represented here. So this would be one of the copies of your DNA, and this is the other copy. And if this was the modification, you see these nucleosomes have these blue modifications, and these ones have this kind of greenish modification, or they are all blue, I don't know. Some of them will inherit the old nucleosomes that contain the modification, but there will be double amount of nucleosomes needed. So there is double amount of histone proteins that are deposited, which we think they do not have the modification because, you know, they are newly assembled nucleosomes. So the old and the new histones, they combine into this old and new DNA. And then the idea is that these parental patterns of histone modifications are reinforced through some enzymes that we call reader-writer complexes. So they basically bind to one chemical modification, and, you know, they have like an arm, let's say, that deposits exactly the same chemical modification in the histone next door. So reader, they read it, and writer, because then they write exactly the same modification next to it. And this is what is used then to kind of preserve these histone modification patterns that we need to preserve cell identity. So I've told you so far that the DNA is organized into nucleosomes. These nucleosomes are made of 8 histone proteins, 107 base pairs of DNA, and a small piece for around 80 base pairs of linker DNA. These histone proteins, they have an N-terminal tail. This terminal tail can be modified with many different types of modifications. And this combination of different modifications makes this idea of the histone code that regulates, among other things, gene expression. And of course, super important, these modifications are inherited by daughter cells. So this is one of the layers of regulation by which, from one and the same DNA sequence, we can create different gene expression patterns. Because what we can have is that one cell has a complete different pattern, genome-wide, of modifications compared to another cell. And by doing that, some of the genes are inactive in one cell type, and those same genes may be active in another cell type. Yeah, thanks to the different chemical modifications of these histone proteins that the DNA is wrapped around. Yeah, is this clear? Good. Yeah, sometimes instead of measuring histone modifications, we measure a proxy of histone modifications, which is open or closed chromatin. So this is again this picture that I showed, where you know, you have this chromatin here, which is very, very well packed. As you can see here, the black line is the DNA. These orange guys are the histone proteins, and these tails here are these histone tails that I was telling you about, and these dots here are these chemical modifications. Now, sometimes, also because measuring these histone modifications, it's quite difficult. Sometimes we just measure whether the DNA is condensed or the DNA is open. Why is that? Let me see, I think I have, yeah. This is just another representation of it. Why is that? This is because this gives us a good proxy for what DNA is active and what DNA is inactive. So if the DNA is extremely packed, this is a thing that we call heterochromatin, it's extremely difficult that anything accesses it. So this DNA will most likely not be replicated, most likely not be transcribed, most likely nothing at all, because it's super packed and nothing can access it. Now, when the DNA is open, it has the potential, at least, of being transcribed. It's a proxy of it. There is a lot of DNA which is open, and still the genes that are not being transcribed. But it gives us a good proxy of it. And I will tell you later on, is much easier to measure than these histone modifications themselves. So it's a concept that has gained a lot of momentum, which is measured this open and closed chromatin. And it's a pretty recent measurement. It's not something that could be done like 10 years ago, let's say. So, you know, it's really now picking a lot of momentum. Now, so until here, I'm telling you about these lila dots here, or these red dots, which are not sitting on the DNA itself, but they are sitting on the proteins that the DNA is associated with. The thing is that the DNA itself can also be chemically modified. And these are these blue dots here, I think. Yeah. So apart from these modifications, these chemical modifications on these histone proteins, the DNA, so really the nucleotides themselves can also be chemically modified. And there is one main DNA modification that we study, which is DNA methylation. And this DNA methylation is deposited in the cytosine. So, you know, we have 4 DNA base pairs, A, C, T, G. And it turns out that actually the C base pair exists in 2 different flavors, let's say. You can have the normal unmodified cytosine, or you can have a modified cytosine that basically has a methyl group that is attached to the 5 carbon of this carbon ring that this cytosine has as a chemical structure, okay? So, yeah, this methylation mark is extremely well studied. It's very important actually for embryonic development, for example, and it's very much correlated to the suppression of gene expression. So in mammalian cells, DNA methylation mainly occurs when we have a C followed by a G nucleotide. So yeah, ACTG. If we have, you know, a CT or a CA nucleotide, we do not have DNA methylation in that position. But if we have a C followed by a G, this C could or could not be methylated. Most likely could, because 70 to 90% of the CpGs in the mammalian genome are methylated. And we have some regions in the genome which we call CpG islands, which is regions that have a lot of these CG combinations in them. These usually are not methylated. And we know that they are generated and maintained by some enzymes which are called DNA methyltransferases. Now, as I told you, DNA methylation, it's a mark which is mainly associated with gene repression. So what does this mean? If you have a position in the genome where there is a gene located, you know, the part upstream of that gene is what we call the promoter of the gene. And it's very important. The state of the promoter of the gene is super important because, first of all, it needs to be open, going back to the histomodification idea of before. If the promoter is open, you can have proteins that bind to that location and that will allow for the transcription of that gene. But at the same time, we know that if the promoter is methylated, so here these lollipops, if you have a black lollipop, this is a methylated cytosine. And if you have an empty lollipop, it's an unmethylated cytosine. So if the promoter, so here you have a gene and this kind of 2000 base pairs, 1500 base pairs upstream of the gene, if that position there is highly methylated, so the cytosines which are in that location are highly methylated, what happens is that it often, this methylation, it interferes with the binding of proteins which are needed to start the transcription of the gene. So in general, when a promoter of a gene is highly methylated, the gene will not be transcribed. What can also happen is that this methylated promoter can be bound by a thing called methyl binding domain proteins. So these are proteins that bind methylation. And this binding will then kind of block, you know, like the promoter is already occupied by these proteins. And since it's already occupied by these proteins, nothing else can go and bind there. So then the gene also cannot be transcribed. Now, for example, DNA methylation has also been associated to phenotypic changes during aging. So you have, sorry, okay. So the top track here would be a young individual and the bottom track is an old individual. What we have observed often is that there are genes in young individuals that have a promoter which is unmethylated And since the promoter is unmethylated, the gene is being transcribed. And what we have observed is that often during aging, some of these promoters, they suddenly become methylated. Since these promoters become methylated, the gene cannot be transcribed anymore. And this happens sometimes, for example, in tumor suppressor genes. So a tumor suppressor gene, it's important that it is active to suppress tumors. And if the promoter of it becomes methylated, then the tumor suppressor gene is not expressed anymore. And we think that this is why, for example, there may be a tendency to develop more tumors during old age. Now, what we have also observed is that we have regions in the genome that we call intergenic. These regions are very repetitive, which means that at the level of the DNA sequence, they have very, very repetitive kind of sequences of letters. And it's super important that we keep these regions of the genome very highly methylated so that all of the Cs in that area are methylated, because otherwise these regions, they are tricky and they start replicating themselves and they start jumping around in the genome and they insert themselves in other places in the genome. So it's very important to keep them highly methylated such that this is not happening. What we have seen is that also in old age, some of these regions tend to lose DNA methylation, and this generates some genomic instabilities also in old age. So this is just to give you an idea that DNA methylation is also important, very important at the level of the phenotype. Now, just to, put it all together, we can look at the DNA as a collection of genes, one next to the other, and we can think of the epigenome as small molecular switches, which are on these genes. So if the switch is on, the gene is on, and I will be making a protein, mRNA, and then a protein. If the switch is off, the gene cannot come on, and I will not be making an mRNA, and I will not be making a protein. These molecular switches are epigenetic, among other things. So it could be that the DNA methylation, which is in that position, makes the gene to be on or off. It could be that the chromatin being very packed or the chromatin very open will allow this gene to be on or off. So the idea is that, when you sample cells from different tissues in your body or in any organism that you're interested in, so you have a bunch of cells that come from different tissues, and every one of these genomes that has these, these genes here, which are depicted with these black and green rectangles. These genes, if you look from a vertical point of view, across tissues, they are all the same. They stay the same. They are in the same location. They code for the same protein. They code for the same mRNA. But in some cases, the switch is off, so there will be nothing being produced. And in some other cases, the switch is on. And we have a lot of genes in the genome. We have like 20,000 genes. So, all this combination of on and off can really have the potential to generate a lot of different cellular phenotypes. And this is regulated by the epigenome. Exactly. So, for example, some of like the most prominent epigenetic, problems. So, for example, fertilization. We have a zygote. The zygote is one unique cell. This one unique cell will divide until it creates a fetus, and it creates then a new individual. So we start with one unique cell. This cell divides, two other cells inherit the same DNA. These two cells divide. We have the 4 cell states. These 4 cells have exactly the same DNA, etc., etc. At some point, these cells start acquiring an identity. And this is epigenetically regulated. And there is a lot of work that is being done, especially like in mouse or in zebrafish or in many other, model organisms to try to understand how is this all regulated? How do we go from one cell, which is called the zygote, to making a whole new organism? Differentiation, hematopoietic stem cells that will give rise to all the different types of cells that we have in blood. This is also regulated by the epigenome. Stem cell maintenance, super important that the epigenome is well regulated there. Cancer, often given by a screwing up of the epigenome that will then allow the cells to, forget about some of the important checkpoints to become replicated and start proliferating like crazy. And we know that the epigenome of older individuals are different from the epigenome of younger individuals. Now there is a big hype on this idea of, transgenerational epigenomic inheritance. If you eat super unhealthy diet, will your children have more chances of having diabetes, for example? Not you, but really like your children. So there is a lot of study also in this direction, where you kind of take one generation, either a female or a male, you do some kind of strong or mild environmental perturbation, call it diet, call it, smoking, stress, something like that. And then you check, you remove then this perturbation, and then you check, is the offspring affected? So have I seen some effect, not just in the generation where I did the perturbation, but also in the offspring? Now, this is obviously tricky because if I perturb a male, the sperm is already inside of it. So yeah, sure, if the offspring of a male is affected, it's because the sperm of the male became affected. But if I go yet another generation and I still see a phenotype, then I could really call it a transgenerational phenotype. In the female, you just need to go one generation further. This in mammals is a very tricky subject. There is a lot of work that is being done in that direction. I know some people, for example, that work in high-fat diet in mice, and then they look at, insulin resistance and so on in the offspring. I think it's super important, and it's, kind of work in progress. I would say there is little known yet about how much transgenerational inheritance there is in mammalian systems. In other systems like plants, it is known that it happens, but in mammalian systems it's a little more tricky. Okay, so this until here was about this molecular basis of the epigenome. So epigenetics is the study of stable changes in gene expressions that are not caused by changes in the DNA sequence. I've told you about the histone modifications. I've told you about open chromatin, DNA methylation. I have not told you about histone variants or non-protein coding RNAs, etc. Now, what is of course important, otherwise we would probably not be talking about it, is that both these histone modifications, open chromatin, and DNA methylation, it can be measured experimentally. Yeah, I mean, otherwise, sure, why are we talking about it? So experimentally speaking, we can actually determine what cytosines are methylated and not methylated. We can determine what histones have certain modifications. And we can also look for the positions of open chromatin in the genome. So I will tell you a little bit how this is done in the lab. So first of all, I will start with how do we do to measure DNA methylation. I'm sure you all know that the genome can be sequenced and resequenced as many times as you want. I sometimes teach this to, more like computer scientists and so on. And sometimes I think, well, better we go back to the basics, So imagine that you have a tissue that you're interested in, some cells that you're interested in. Imagine that you have some human cells that you're interested in. I told you that the human genome has been sequenced, but what does it mean that the human genome has been sequenced? It means that we have the reference letters of the human genome, but I don't know your specific human genome. If I want to know your specific human genome, I need to extract your cells and resequence them. So the way it works is that we take cells that we are interested in. This, I think from the picture, it was some kind of bacterial cells. We can extract DNA from a lot of these cells together. And then this DNA, we do DNA shearing. So we kind of, cut it into small pieces. So this is what's here. DNA is cut into short fragments of known length. Then we have these small pieces of DNA, and we can PCR amplify it. Now we all learn what PCR is. Before Corona, few people knew what PCR was. So now, thanks to PCR, we can make a lot of copies from a piece of DNA. Why do we do that? We do that because in general, when we do this procedure from cells to, these pieces of DNA, we do not have a lot of material. And what we want to do is that we want to read the letters that are sitting on that material. And for that, we need a lot of it. So we just PCR amplify it such that we make more copies of it. So we PCR amplified, and then we do what is called DNA library preparation and DNA library sequencing. What this means, at least for me, I'm a physicist, what this means is that I have these pieces of DNA. These pieces of DNA have nucleotides sitting on them in a given order. And what the sequencer does is that it basically tells me for every one of these pieces of DNA that I put into the machine, what is the exact order of the nucleotides on that position. So what I get out of the machine is a bunch of what we call reads, because we kind of say, you know, we are reading the DNA. So these reads that tell me from beginning to end what letters were on those locations. And what we usually do is that then we blast these reads. So these reads have, I don't know, let's say 150 letters on them in a given order. And then I blast it and I ask, okay, these 150 letters, where does it come from in the whole 3 billion base pairs of the human genome? And since I have like 150 of them, they usually only come from one position. So there is only one position where I would say like, aha, these letters, they came exactly from that region in the DNA. And I can do that because I know the reference genome. Now, what's the trick here? We're talking about the epigenome. We are not talking about the genome. The trick here is that there is a thing called bisulfite treatment. And bisulfite treatment uses sodium bisulfite. And sodium bisulfite is a chemical compound that takes unmethylated cytosines and will convert them into uracil. So we don't really care about the specific, you know, chemical reaction that will happen. But what's important is that if the cytosine was not methylated, so it didn't have this appendix here, remember, where there was a modification here, this one doesn't have it, it will go through, like, you know, a bunch of chemical reactions that will convert this guy, which is a cytosine, to this guy here, which is a uracil. And then the uracil, by base pairing, it will get converted into a thymine. So effectively, what I do is that if, you know, like at this stage here, I add sodium bisulfite to my mix. Basically, what I do is that I change the Cs, which did not have the chemical modification, I change them into Ts. The Cs that had the chemical modification will not get modified. They will stay as a T. So what it means is that if I know the true sequence, because, you know, I have my reference genome, like the human genome, I know, you know, I don't know, chromosome 2, position 10,000, I know what's the true sequence sitting on that location. Then when I look at my bisulfite converted sequence at that location, what I will see is that if I see a C after bisulfite conversion, this will mean that the C was protected by a methyl group. But if I see a U or a T at the position where there should be a C, this means that this C was not protected by a methyl group, so this C was unmethylated. Now, this is how it looks like. This is like an example. It's obviously a toy example. This is a bunch of letters on the DNA, and every one of these green lines here represents one of these reads. They are in general like 100, 150. You can have them as short as 25 base pair, but that's a little bit more tricky than to know where they come from. So let's say they are 100, 150 base pairs long. These reads from, you know, from green to green. Here for the toy model, I made them much shorter. Every time that I have a cytosine, I can look into my read and I can count how many of these pieces of information that come supposedly from exactly the same location of the genome, how many of them, they give me information that this cytosine should be methylated, and how many of them tell me that this cytosine is actually not methylated. Yeah, so I have situations where it's pretty clear. I accumulate a lot of reads at a given cytosine in the genome, and all of the reads tell me, yes, there was methylation there. So yeah, most likely the cytosine is methylated. There are positions where it's also super clear that it is unmethylated. There are the majority of the cases, isn't it, where the position is kind of intermediately methylated. Now, from a data analysis point of view, I will not go at all into the details, but what we can do is that for every one of these locations, we can determine a given methylation level. And the methylation level is going to be how many instances I measured that told me that there was methylation in that location, divided by all the instances that I calculated at that location. So a cytosine like that would correspond to methylation level 1. So this position is 100% methylated. Everything that I measured told me there was methylation. A cytosine like that would correspond to a 0. So all the evidence I have is that there is no methylation at all in that position. A cytosine like this one would correspond to 0.2, which would be here. And in general, when we take all methylations in the all cytosines in the genome, and we look for these methylation levels for any given tissue that we have measured, we in general see a distribution like that, sometimes with a little bit of a bump in the middle at the intermediate methylation level. There are then different algorithms that people use. What is the right cytosine methylation? What do you mean? What is the right cytosine methylated? No CG. Yeah, because I made a mistake on my skin. Good that you caught it. I will change it next time. Thank you. So yeah, the thing is that there are kind of, statistical tests that we do then to not be super crude and say, well, more than 0.5, it's methylated, less than 0.5, it's unmethylated. In general, like you will sequence your sample in a given way, that you will know what is your error rate, how much methylation I observe when it's not methylated, how much unmethylated I observe when it's methylated. And then you use this error rate to decide, how likely it is that I observe 2 reads out of 10 being methylated or not. But yeah, the idea is that basically, when I have a low methylation level, I will say the cytosine is unmethylated. When I have a high methylation level, I will say the cytosine is methylated. And the beautiful thing is that thanks to this bisulfite sequencing, we really can go into any tissue of interest, any cell type of interest, and really get a complete genome-wide characterization where basically, like for every cytosine in the genome, we can determine whether it's methylated or unmethylated. Yeah, I will talk about single cell later on. When you sequence a single cell instead of sequencing a tissue, how many copies of a chromosome do you have in a single cell? You have 2, which means that, sure, you can PCR amplify it a lot, but you will PCR amplify exactly the same 2 copies. So at the end of the day, you only have 2 copies. And it's very, very likely that you will not be able to recover one of the 2 copies because, when you do bisulfite sequencing, you need to wash your sample to take the bisulfite away. When you wash it, some reads will also be washed away. You only have 2 copies. Yeah, as soon as you wash a few reads away, you wash a lot of information. So this is more the typical situation in a single cell experiment, where maximum per cytosine, you will have 1 read covering it. And this read will either be methylated or unmethylated. And that's all you can do. There is just, from a technical point of view, you just cannot recover more than that. Now, how do we do to measure histone modifications? We actually do a complete different thing than when we measure DNA methylation, and we do a thing which is called ChIP-seq, chromatin immunoprecipitation, followed by sequencing. What does this mean? So you take your organism of interest and you take the cell types that you're interested in. You have a single cell suspension. And if you remember, I told you, you have this DNA, which is wrapped around these histone proteins. And the histone proteins have these tails that have these modifications. So what you do is that when you, remember I told you when you sequence the DNA, you first cut it into small pieces. Here, instead of just cutting it randomly into small pieces, what you do is that you do a digestion with a given enzyme that cuts in between these nucleosomes. Yeah, so I had this, remember this microscope image where you saw these pearls, let's say, with the DNA in between. It really cuts in between the pearls. And then you really have these histone proteins with the DNA wrapped around and the modification. So some of these proteins will have different modifications on them. And then what you do is that you have antibodies that will recognize only one type of modification. So an antibody binds to this green modification, but does not bind to anything else. So what I can do then is that I can separate the part of the chromatin that was bound by the antibody from the part of the chromatin that was not bound from the antibody. So I only have left here the part that was bound. And then I will do DNA sequencing, normal DNA sequencing, but only of this part here. So what this means, and sorry for the super low resolution of this picture, where I need to move your faces away. If this is, these groups of proteins and the DNA wrapped around, here, imagine that I have an antibody that recognizes only this orange modification. So I will only sequence these orange reads, and the rest of the genome I will not sequence at all. So when I recover my DNA and I ask myself, where do these reads come from the whole genome? And I, ask myself, do they come from here, from there, from there? What I will find is that they only come from those positions where the modification was present. So ideally, what we have is that only the DNA that was wrapped around these modified histones that were bound to the protein of interest, they are sequenced and mapped to the reference gene. So in principle, here I will have nothing mapping, and suddenly, I will have a lot of reads in that location. Then there will be nothing mapping, and then I will have a lot of reads in that location. So it's different from DNA sequencing, DNA methylation sequencing, because in both cases, we sequence the DNA. But in the case of DNA methylation, we really need to look at the information of the reads, because whether there was a T or a C will tell me whether it was methylated or unmethylated. Here, I actually don't really care about the information that is on the reads. I only care about where are the reads mapping. And this is how it looks like in reality. So what I usually see in these experiments is that I have like a low level of background noise, kind of reads mapping more or less everywhere in the genome. And then in some locations, I clearly see that, whoa, I have a lot of reads that are mapping in that location. Yeah, so here, this is kind of a condensed view where I only kind of, plot the number of reads that are mapping in a given location in the genome. And here, yeah, from an analysis point of view, which is what I, coming from the computational world, what is the goal here is to determine the enrichment above this background. In some cases, it's super clear because there is a very low background and huge signal. In some other cases, like this example here at the bottom, is a little bit more tricky because, the signal to noise ratio is much worse. Yeah, I had an example here, but I mean, we don't really want to go into that. Yeah, so now I will go into the last thing that we can measure, which is the open chromatin. I told you this is a much easier assay to do in the lab. This is a much easier assay to do, among other things, because I didn't say that, but when you measure histone modifications, this is actually a very tricky assay to do in the lab. And it has to do, among other things, because you need to have these antibodies that recognize specific histone modification. And these, they often have a lot of off targets. So it's a little bit tough. Sometimes you get a lot of background noise. Sometimes you don't really know if your signal is a true signal or is these are the off targets and so on. And the other tricky thing is that you need to measure a very long panel of different modifications to get back to this histone code that I was telling you about. You would need to measure, I don't know, methylation on the lysine 3 of the histone, lysine 27 of the histone 3, and methylation on the histone 4 at position whatever. And, so you really need to measure a lot of different things to be able to more or less get back to this histone code. The idea of doing this, what it's called this ataxic, open and closed chromatin, is that you don't really care what are the modifications, but you just want to know if the DNA was open or it was closed. And so how do we do that? This is a closed chromatin here on the left. This is open chromatin here on the right. There is a thing which is called a TN5 transposase. This, what it does, so it's an enzyme that it actually cuts DNA. And it can only cut the DNA if the DNA is open. So whenever I have this, very condensed DNA, it just cannot access it. But if the DNA is open, it really can cut it. And what people have done is that they have engineered this TN5 that have what's called primers on every side. So in this scheme here, it would have cut in this location, cut in this location, in this location. It will not have cut here. And after it cuts, you see that in between the black DNA here and the black DNA here, I have a red and a blue piece of DNA. So it cuts and it inserts this red part and this blue part. And then here again, it has cut and it has inserted the red on one side and the blue on the other side. Sorry. Then what we can do when we sequence this is that we do not sequence everywhere in the genome. We only sequence the positions in the genome that start with this red sequence or start with this blue sequence. So again, we do not sequence anything else. We only use these primers to start the sequencing reaction. And then what happens is that in principle, whenever it was closed, there was nothing to be sequenced. And whenever it was open, I will again see a pileup of reads that indicates me that that position in the genome was accessible by the TN5, and therefore it could be sequenced. And I also have, again, these peaks along the genome. Yeah, exactly. So this is just to say when it's closed, I have nothing. And when it's open, I have these peaks of what is called chromatin accessibility. Again, single cell, super tricky when we do this assay per single cell. Again, we are measuring the DNA. We have only 2 copies of DNA per cell. We want to access the open DNA of a single cell. So the data is extremely sparse, and we will never see a peak like that. We are lucky if we see 1, 2 maximum number of reads that map to any location in the genome. Yeah, so okay, this is just to wrap up. The idea here is that, sorry, move your faces again. So in general, let's say from a classical point of view, not single cell point of view, I have many different cell types, many different tissues, many different organs. And what I can do is that, for many of them, I can basically, decide on a panel of epigenetic marks that I am interested in. I measure them. This is like a snapshot of a position in the genome, like this x-axis would be really like the position in the genome. Here in blue would be the genes that fall in that location. And then I have, all of these patterns that will tell me, look, there is I don't know, like a peak here, and there is a peak there. So this gene is most likely transcribed, and so on and so forth. Now, I worked in my group. I'm developing algorithms for the analysis of single cell epigenomics and not bulk epigenomics. And I wanted to give you a little bit of an idea of what that is. Oh, wait, there is a question. I don't really see the chat very often. So if I don't answer, you just shout. How do you make sure that you have adapter A on the one side and adapter B on the other side? What if both sides are inserted with the same adapter? Good question. So if both parts are inserted with the same adapter. So I know that when you cut, the 2 adapters that you add are different. But in any case, that shouldn't really matter, isn't it? So after you have put your adapters and you have your reads that will have, the adapter in them, you again amplify them. And I guess it doesn't matter whether you had one or the other adapter. In general, what you see, and this is the part that I didn't go to during the analysis, the slides that I skipped, what you usually see is that you never really see one peak, but if you sequence very deep and you have a lot of resolution, you see like 2 peaks, one next to the other, because in one case you sequence in that direction and in the other case you have sequence in the other direction. This happens both in ChIP-seq and in ATAC-seq. So the typical algorithm that analyzes this kind of data, it starts by doing like a shift of these peaks to kind of put them on top of each other. This is an algorithm which is called MAX2. I'm honestly not so sure about this question. I'm sorry about that. We could ask, if you're interested, you can follow up and I can ask someone that does the experiments to make sure we understand it. Sorry about that. Good. So single cell epigenomics. Yeah, there are many different things now that can be measured at the single cell level. I don't do the experiments, as you just saw. It's really, really tricky to do all of these sequencing experiments at the single cell level. A single cell has a tiny amount of DNA. So if you were to use any of the usual sequencing experiments on a single cell, you wouldn't get anything out. Bisulfite treatment, the one that I told you about, that converts the C's into T's, If you leave it for a little bit too long, it basically eats up all of your DNA. So you need to leave it for a little bit and then you wash it out. Wash it out, it's fine in bulk. You have a lot of cells, you can wash it out. And if part of your DNA is washed out, doesn't matter, you have a lot of DNA. If you do it for a single cell and you need to wash out, every time that you have a washing step, you know that you will get rid of a big part of your DNA. And you only have one cell. As soon as you get rid of the 2 copies that you had there, that's it. You've lost that part of your cell. So all of these protocols that you see listed here, this is a little bit of an old review. So it's not dramatically different at the moment, but I would say these protocols have been optimized a lot since then. What these protocols have been very, very good at is at, first of all, miniaturizing the reactions, such that we need very, very little amount of reagents because it's just one single cell, and getting rid of all the washing out and all the pipetting and so on, because, you know, every time you pipette, your DNA actually kind of sticks to your pipette, and then you recover less and less of it every time. So they have really gone to the point where you do the minimum number of steps. And the most, I would say, some of the most used and successful methods that people use at the moment are droplet-based. I should have put a video here because I find it amazing. Droplet-based means that people have engineered really like nano droplets that inside of the droplet have all of the reagents that you need to construct one of these libraries that then you put into the sequencer. And then you get a microfluidic device that pumps one droplet and puts one cell inside of the droplet. And then it pumps another droplet and puts one cell inside of the droplet. So it's extremely miniaturized. All the reagents that you need are already inside of that droplet. And you load your cells in it. And what comes out of it, it's a library where every single cell has been barcoded. So then you can get back on your computer what cell was what. So these are, let's say, some of the most successful methods at the moment. We can measure many different things. I do not particularly like this plot of a single cell, but we can measure histomodifications doing single cell ChIP-seq. It's totally unsuccessful, so very little people do it. We can measure DNA modifications mainly with the single cell bisulfite sequencing, so we can know what cytosines are methylated or not. The most used method is definitely the single cell RNA-seq, where we do not really care about these layers of regulation, but we only go into already what's a transcriptome like for any single cell. This method is obviously most successful because from one copy of DNA, you make a lot of copies of RNA. So you have a little bit more material to sequence than when you sequence the DNA, for which you only have 2 copies. You can also measure chromatin organization, so like a 3D context with a single cell Hi-C. And you can also measure DNA accessibility, mainly with the single cell ATAC-seq, which is the single cell version of the ATAC-seq that I told you about. So there is a strong data sparsity, extremely strong data sparsity, but we compensate this strong data sparsity by measuring a large number of cells. So from, it's kind of a little bit of a you need to shift your comprehension, like in bulk, in general, you were getting one tissue and you were, for that one tissue or that one cell type, you had a lot of different experiments that you were doing. Here, what you have instead is that you have a lot of cells, like 10,000 cells, 1 million cells, for which you have measured only one experiment. So in this case, it would be, for example, these lollipops with DNA methylation. So, you have like for cell 1 until cell 1 million, the positions in the genome. Yeah, so it kind of changes from one tissue doing a lot of assays to actually doing one assay into a lot of different cells because there is a lot of sparsity in your data. So I will tell you a little bit what we have done in the lab, in my group. So the first thing that we do when we analyze this data like that, so, we have a lot of cells, and we have for every one of the cells their genome with some epigenomic information in them. So the first thing that we do is that we do what's called feature selection. Why do we do that? Because if we have 1 million cells in this direction, and we have, for example, the DNA methylation status of every cytosine in the genome, my computer will say, no, I'm not going to do that because this is way too big, okay, from a dimensionality point of view. So what we do first is that we decide, okay, what features in the genome we want to focus on. This is just to say, if you do the single cell transcriptomics, you will just look at your genes because that's the only place that you have information from. The beauty of the epigenome is that you can actually look at other positions than the genes because you have information from everywhere in the genome. Now, the way that we have approached this problem is that we've basically created a package that does single cell epigenomic data analysis in Python. And we allow for this data to be analyzed by using many types of different feature spaces. So you can go by windows. You say, I don't know anything about my cells. I just tell you 5 kb windows and you tell me how much DNA methylation there is in that 5 kb window, and then the next, and then the next. Or you can say, no, come on, I know that, DNA methylation at promoters is really important. So I will choose to only look at the DNA methylation at the promoters. Or you may say, no, for my specific tissue, I don't know, I have neurons, so I know that the DNA methylation in the gene bodies is much more important. So I look at DNA methylation in the gene bodies. Or I look at DNA methylation at transcription factor binding sites, because as I told you, if they are methylated, the things cannot bind. If they are unmethylated, the things can bind. So, you can really look at many different positions in the genome, but in a more biased way, where you really use the annotation and the knowledge that we have about the genome. If you have open chromatin, as I told you with a taxic, you have these peaks, isn't it, in the genome? So in general, what we do is that we have sequenced every single cell separately, but in the computer, we put all the reads together as if they were coming from one and the same experiment. And we do this thing that we look at where are these peaks and when there is background, and where are these peaks and where there is background. And then we have this position of the peaks, that would be these positions here. And then for every single cell, we only score how much open or close are these peaks in my single cells. So basically what I do, I have these different tracks here, which for the ones of you that work with computers, these would be bet files. And then I will just, superimpose the bet files onto my cytosine methylation data or open data. And I end up with having a bunch of matrices. And my matrices have every row is one of the single cells, and every column is a summary of what was the status of the epigenome in these given positions of the genome that I've decided through my features. So windows, for example, or enhancers or promoters, peaks, transcription factor binding sites. This pipeline is called EpiScanPy, epigenomic single cell analysis in Python. Once you have decided on the features that you want to use, which from an analysis point of view, it's as easy as providing a bet file. A bet file is just a list of starts and stops of the genome. So we call it feature selection, but you just provide me with a bunch of features. We, of course, need to do some pre-processing. We kind of clean all the dirt, all the cells that are not covered, all the peaks that there is really no information anywhere, and so on and so forth. So we reduced a little bit the number of cells, we reduced the number of features by just keeping the most variable, the most covered, etc. And what I do have, sorry, I won't forget your faces. What I do have is a thing that is very fancily called neighborhood graph, but actually everything that it is a matrix that in every row has a single cell, in every column has a feature, and it has a quantification of that feature for that particular single cell. Now, what I can do with this fancily called, with this matrix, this is the graph, is that I can use this as a vector, and I can calculate the distance between any pair of cells in my population. And this is what it's plotted here. This is the graph. So obviously, one cell is most similar to itself. So this is the diagonal. But then I will start already see some groups, of cells that are much more similar among each other, and some cells that are more dissimilar among each other. Now, when I have this quantified like that, from these I can construct what's called a KNN graph that will put an edge between two cells if this value here is higher than a given threshold. Yeah, so now every cell is a dot, and I will put an edge or no edge, depending on a threshold or a given number of neighbors. I usually say, for example, I want 50 neighbors. So every cell will be connected to 50 neighbors and will be disconnected to everything else. And now that I have this in the form of a graph, then I can do a lot of stuff with it. For example, I can do a low dimensional representation of my data. Here, this dimension is what we call UMAP dimensions. Every one of these dots is a single cell. And the cells which are more closer to each other in these 2D dimensions, they were also closer to each other in this 500,000 dimensions matrix that I showed you initially. I can also, oops, we don't care about that. This is too detailed. I can also color these cells by groups, and I can tell you, look, these cells here, they seem to really be very, very close to each other compared to anything else. So I can look for clusters of cells. And then I can know what these cells are by doing differential calling, which will allow me to do annotation. I will give you examples of all of that, pseudotemporal ordering or data integration. So just to make it more concrete, I will give you two short examples, one on single cell DNA methylation and the other one on single cell open chromatin data. Both cases is with publicly available data. So the single cell DNA methylation is frontal cortex, so brain of mice, single nucleus methyl C sequencing. I have more than 3,000 cells, it's not a lot. As I told you, the data is extremely sparse. What does it mean, extremely sparse? In this case, it means that from these mice, from all the possible cytosines in the genome, less than 5% were sequenced in any one of the single cells. Yeah, so tiny, tiny, tiny amount of information. Despite that, I still can get a methylation status of every one of the cytosines that has been covered. And yeah, I mean, this is just to remind you, this is what was at the beginning of the lecture. And I can build count matrices using different annotations of the genome. So yeah, you know, like enhancer, promoters, windows, you know, gene bodies. And then, as I was telling you, once I have this kind of graph representation, I can go into low dimensional representations. And then I can ask myself what cells belong to one cluster, and I can paint them in that color. So I can do that using different representations of the genome. What you see here in this, so what does this two-dimensional plot mean? Every dot is a single cell. These are these 3,000 neuronal cells. These are exactly the same 3,000 neuronal cells. But here, when I constructed this matrix, where I had all the cells and all the columns that were positions in the genome, the positions in the genome that I asked what's the methylation level of were promoters. In this case here, I instead did 5 kb windows. So I took all of the genome, and for every 5,000 base pair window, I asked for the mean methylation level of all of the cytosines that were in that location for every single cell. In this case, I also scored DNA methylation levels, but at enhancers. And in this case, I scored them at gene bodies. So every dot is a single cell, and they are colored by a method which is called Louvain clustering that tells me what cells most likely come from the same group. So from this graph point of view, you know, like what cells have a lot of connections to the cells in the same group, but actually have very little connections to the cells which are outside of that group. Now, sorry that things moved around. So these are the promoters, these are the enhancers. And what I can do now with this kind of representation is that I can start to ask questions. These numbers here are the clusters from the promoters, and this number here are the clusters for the enhancers. And I can already, for example, identify I have a cluster in the promoter, cluster number 12, that it's split into 2 clusters for the enhancers. These are the same cells, okay? So these same cells here that are split into 2, in the promoters, they are only called one and the same cluster. So what I can interestingly see is that when I look at different parts of the genome, I will find slightly different grouping of my cells, only slightly, okay? In general, the information is the same everywhere. But in this case, because it's a publicly available data set, I don't really care. But if this was my data and I had some particular questions with it, of course, one can then dig very deeply. You know, why were the enhancers important to splitting these cell types into 2? While if I only look at the promoters, I couldn't have been able to tell the difference, for example. And what I also see, sorry again that things moved around, is that, for example, the enhancer representation, as you can see, like you see these groups of cells, they are really tight. They, like, you know, the gene body is the worst, really, where, like, you know, like, yeah, you really cannot tell the difference. So you see that some genomic spaces, like enhancers, seem to give you much more information about the cell types than, for example, windows or even promoters. And this you can quantify it computationally speaking with a thing called the silhouette score. Now, okay, this is great, but here these are cells with colors, and probably you don't care about cells with colors. You want to know what are these cells. So to know what the cells are, what we do is that we identify markers, which means that we rank differentially methylated features. So we can do that in any of the feature spaces, but promoters is the easiest because the promoter is right in front of the gene. So it's kind of easier for me to interpret. What do? Here is just an example. So I show you the same cells as before. You see the cells are the same ones, but here I had clustered with a lot of resolution. I had asked to really find a lot of different cell types. Now here I just say very low resolution. So I only get the two biggest clusters in my data. And I ask, what features in the genome? Remember, I have my cells and I have my columns with features. Now I ask, between these two clusters, the orange and the blue, which ones are the columns in the genome which are the most different between these two clusters? And this is what I get as a result. So these are different promoters of genes, and this is their methylation level. And when I look into the list, I see that one of the genes which is differentially methylated is the NeuROT2. This representation is not super clear because there is a lot of missing data. So what I will plot it instead is that I will plot the methylation level of this promoter of this gene NeuROT2 on top of every dot. So every dot was a cell, and now I'm plotting on top of every cell the methylation level of that specific promoter. So in some cases it's high and in some cases it's low. I can also plot a violin plot. Yeah, now this is a distribution of all the blue cells and all the orange cells. Now, since I talk to my biology collaborations and they tell me NeuROT2 is a marker for inhibitory neurons, then I'm like, aha, okay, so interesting. We found two big groups. One is the inhibitory and the other one is the excitatory neurons. And I can dig them deeper and deeper and deeper inside of these groups. And for, you know, I can compare every small group to all the rest. Groups that are very close to each other, I can compare them to each other and so on. Every time I find a list of dispositions that were the most differential, I can then find a list of genes. And then most likely with, you know, in collaboration with biologists, then we can be like, aha, so these were the most likely cell types that I know have these genes being upregulated, for example. Now, we can do exactly the same thing with open chromatin. This is another example, again, publicly available data, and this is blood. Blood is very interesting because it's differentiating tissue. So, you know, the cells constantly go from hematopoietic stem cells to fully differentiated cells. And this is a paper for which they had sequenced more than 60,000 blood cells doing single cell ataxic. I told you the data was very sparse. Indeed, in that data set, on average, per cell, we only have 13,000 reads. So per cell, we only have 13,000 data points, you know, along the genome. So it's really, again, very, very sparse. What I do, exactly as with DNA methylation, is just that here, instead of quantifying methylation levels, I'm quantifying open levels. I quantify how much every cell is open in every peak. And I do again this low dimensional data representation and clustering. So this is what I find in this case. Disclaimer, I already put the cell type names here. How do I found these cell types? Well, what I did is that for every one of these cell groups at that moment, I didn't know that there would be B cells and B cells, whatever, T cells and so on. What I did is that I asked again, when I compare B cells to all of the rest of the groups, are there some peaks that are very open in those cells and very close in the others? So this is, you see openness. So this is the most open possible. This is the most close possible. And what you see is that these two peaks here that have been numbered 30,816 peak and the peak 47,657, they are super open in the B cells and they are completely closed in the other ones. Same for these 2 peaks, same for these peaks. Of course, when I do cell type identification, I do not look at 2 peaks, I look like at the top 100, but this is the idea. And then we have developed a small function that for every peak, it tells me, you know, what's the closest gene to it, so I can start doing cell type identification. And I can come up with these nice names here. Now, I told you, blood is a differentiating tissue, which means that actually I shouldn't really be looking for groups of cells, isn't it? Because groups of cells in the neurons, it kind of made sense. These are fully differentiated cells, terminally differentiated. So I have different types of cells. Now here, this is more fluid because, well, I mean, the progenitors, they somehow still give rise to everything else. So looking for these clusters is not the best way. And what we do in this case is that we have a concept which is called pseudotime. And what pseudotime does is that instead of looking for groups, it kind of instead orders the cells based on their similarity. And then you kind of root your pseudotime. In this case, we rooted it in the progenitors. And then you get this representation on a different dimension, which is called dimension component, diffusion component. And what we could see here is that the progenitor cells gave rise to all of these different cell types. And, you know, then you can basically look at the different projections and you can follow how, you know, one cell type, it kind of bifurcates at some point and it gives you different cell types and so on. Yeah, so this is just what I have presented about Episcampi, which is this pipeline that we have generated, but mainly the idea here was not so much to sell, you know, our pipeline, but to give you an idea of all the cool things that can be done by studying the epigenomes of single cells. Just one last slide, because this is where the field is going, and I just wanted to give you a feeling. The field now is going into the single cell multiomics. So, you know, the DNA content in single cells is tiny, but what we are now able to do, not me, what other people are now able to do, is that for one and the same single cells, they can sometimes measure many of these layers. So it's possible now, for example, for one and the same single cell to measure both the chromatin architecture and the transcriptome. So now for one single cell, huge amount of missing data, but for one and the same single cell, I can determine what positions in the genome are open and what genes are being transcribed. Now, again, with single cell, there is very little that you can do with one single cell. But if you measure a whole population, and then you can start, you know, grouping the cells by their similarity, now you really have truly paired data, where for exactly the same cell, you know what genes are transcribed and what chromatin is open or closed, what pieces of the DNA is methylated or not. So this gives us a much better resolution into this idea of studying how do these epigenetic mechanisms allow me to go from one and the same DNA to so many different phenotypes. Here, the phenotype, let's say, is the expression of the genes. And I can measure the epigenome. I know that the DNA is the same. This is the same organism, the same human that I have profiled. So this type of data now has so much potential for starting to answer a lot of these epigenetic questions that we have been trying to answer for so many years. It has a lot of challenges, is extremely sparse. You need a lot of single cells. It's really what we call big data. But I think it really has a lot of potential for answering a lot of epigenomic questions. And yeah, I think this was just called the method of the year 2019, single cell multiomics. And this is again my group, and this is the end of my presentation. And if you have questions, you can ask them now. Thank you. Do you have questions? So there is one question, but Andreas, do you want to unmute yourself? It's maybe nicer. Okay, good point. Yeah, so CPG islands indeed are very important for the regulation of gene expression. So yeah, if CPG islands are located in promoters and these are, you know, highly methylated or not highly methylated, they will of course regulate the expression of the genes. The concept of CPG islands is very prevalent in mammalian genomes. So often a lot of people, when they measure DNA in mammals, they do not really measure it genome-wide, but they only look at CPG islands. For the people here that I know that are working in plants, there is no such a concept as a CPG island. And there the DNA methylation is kind of much more uniformly deposited in the genome. But yeah, definitely.
Maybe I have one question
Sure.
about intergenic methylation, and also methylation in intergenic regions, because you mentioned that it could affect cancerogenesis, gene expression, cell transformation, etc. So I would like to ask to a subtelomeric, because I know that telomeric regions cannot be methylated, but subtelomeric can be, and if it could affect the stability of telomeres and cell transformation as the effect of that.
That's a great question that I have unfortunately no answer for, sorry, because I guess I'm not a biologist. I know that DNA methylation in transposable elements is super important, you know, to prevent them from jumping around, transcribing themselves. Subtelomeric regions, I have no idea. They are repetitive regions, I assume.
Exactly, that's what I was thinking about, but you mentioned about this repetitive regions, so subtelomeric regions can have this pattern,
yeah,
as in between like telomeres, but maybe it could affect also but other factors. Maybe there are some RNA inferencing, I don't know.
Do you know if they are heterochromatic? Are they open or closed at the level of heterochromatin? Do you know?
Normally they are closed, I guess, but if they just get open, something could happen. But I don't know either. I read one publication in 2017, and it's pretty old one about cancerogenesis in stem cells, cancer stem cells, something like that. And they put this hypothesis. So I was wondering if there's something new in this field. But maybe I will just go to literature and find out.
Yeah, sorry, I don't know about it. I know that, of course, you know, telomeres, as you were mentioning, it's also super studied in cancer and in aging, you know, but just because they get shorter, not so much for their epigenetic regulation per se.
And also I have, sorry. I have also one question, if I may ask, about the single cell multiomics, because you mentioned that the biggest problem would be the missing data. Are there some algorithms developed right now, or they are just up-to-date algorithms which are make this imputation of the missing data, or it's still a novel thing that nobody touched it?
Yeah, no, this is a very active. So to be honest, when the field started, there was a lot of emphasis on imputation of missing data. And you have some algorithms that do that for single cell RNA-seq. I don't really remember the names. One is called Magic, I think. And so they try to impute the missing values. Where the field, I would say probably it's now going more towards, is to generate huge amounts of cells. So if instead of having 500 cells, which is how the initial studies started, you have now 1 million cells, you start having less of a problem with the missing data, because every cluster that you will, even if you want to really subcluster to find a very rare cell type that there are very little cells of, you will still recover at least 10, 30, 50 cells of that given cell type. So in general, when you will, you know, aggregate them on the computer or start doing all of these tests that I was telling you about, the missing data becomes less and less of a problem. For the more computational people here, one of the methods that people use is these autoencoders, where, you know, the idea is that, so the autoencoder, what it tries to do is that it tries to reconstruct your input. And some people are using that for reconstructing your input without missing data. So yeah, this is a very active field. Some people also use it, for example, for doing batch correction, because we obviously didn't talk about that. But you know, there is some of these super nice initiatives like the Human Cell Atlas. The goal of the Human Cell Atlas is to get, you know, the same way as you have the periodic table with every chemical element, you would have a periodic table of the cell types where you would really be able to super well characterize every single cell type that exists in the human. And there are a lot of groups in the world that are working on that. And now I lost my train of thoughts. Yeah, so these are data sets which are produced in different labs. They come from different patients. They have, different genotypes. So when you do one of these plots that I was showing you about, where you try to identify cell types, it looks beautiful. But then if you color your groups, they belong to the lab where the experiment has been done, or they belong to the patient that the samples come from. So there is also a lot of work on doing this kind of batch correction. People also use similar techniques like, either variational autoencoders, where you put the patient or lab of origin as one of the covariates or things like that. So, yeah.
Thank you very much.
Thanks. Good. So if there are no more questions, thank you very much for your time. And yeah, hope to see you maybe in person, some of you that I know at least soon. Bye-bye.
Thank you.